happened in Nice, which was really a great meeting, extremely interesting. So now we give the floor to Andy Miller, who is a very well-known professor of psychiatry at the Emory University in the States, and who, as you know, is a world expert in the field of brain-immune interactions, in particular in the field of depression. And so he's going to describe lessons learned from failures. So not to feel depressed, but to raise hopes on how we should fight them. So Andy, thank you very much.
Thank you, Marianne, and welcome everyone. This is really a treat, and thanks for making the effort to come here in person, and thanks to all of the people who are online. I don't know what to say. I think I feel like I'm like the ghost of Christmas past here. I'm going to talk a little bit about what we did in the first Precision Psychiatry meeting, which was held, as Marianne mentioned, in Switzerland, in Crans, Montana. And that was the Precision Psychiatry think tank, I think is what we called it. And that was the first meeting. And I think it's important to keep in mind that there are a tremendous number of opportunities in precision psychiatry, but there are also considerable costs if we don't do it. So this is not necessarily a choice in that respect. And one of the things that I presented in the meeting that we had last time followed a meeting that was focused largely on the impact of inflammation on the brain as it relates to depression. So much of what we were talking about. Oh, you don't have it on slide mode, is what it is. Yeah. You got it now. You want me to go back? Okay. Sorry for those of you online. I guess we saw it here. Uh-oh. Is it going by itself? That's scary. It's a crisis. Anyway, so there was a large focus on the immune system and its effects on the brain as it relates to various psychiatric disorders and depression in particular. And I sort of got started in this whole area of precision psychiatry because I'm kind of off doing my own research having to do with inflammation in the brain. And I began to read the literature and realized, at least in my mind, that there's a crisis ahead. And the problem is the clinical trials. And in particular, I was worried and concerned about the clinical trials that have been conducted with anti-inflammatory drugs in depression. And I think many of you are aware that there have been a number of trials, but 30, 50, depending on what you want to count. And the meta-analyses of this literature suggest that anti-inflammatory drugs may have some efficacy in treating depression. However, the results have been inconsistent and largely underwhelming. So they're not pharmaceutical companies racing out to develop anti-inflammatory drugs to treat any psychiatric disorder at this point. What really concerned me were these more recent trials, two large recent trials, the Hellman-Regan et al. Trial in 2022, Hussein et al. Trial in 2020, large samples of subjects using two of the major drugs that are represented in these meta-analyses, celecoxib and minocycline, and both found no separation from placebo. This is what the data looks like from the Hussein et al. This is published high-profile journal here. Minocycline, these are adjunct treatments. Celecoxib, minocycline, the combination compared to placebo, and you can see there's absolutely no difference between the groups. And then you go on and you read the conclusions in the abstract, which is what most people read. And yeah, I get it. No evidence that minocycline or celecoxib separated from placebo. But it was this last line that really drove me into this space. And it says, this large trial casts doubt on the potential therapeutic benefits, you can see, of adjunctive anti-inflammatory treatments, drugs for the management of depression, bipolar depression in this case. That is a field killer. And if we don't pay attention, this field is going to die. And the problems are obvious, and they relate to what both Martine and Marianne were alluding to, is that the way we're conducting clinical trials now in psychiatry are legacies of the past. They're born out of a series of very non-specific drugs with broad spectrum activity that we all use for all disorders. We use antipsychotics for everything. We use antidepressants for everything. And so it really doesn't matter how you can conduct the trial. If you just get depressed, it'll get better on certain antidepressants, and we'll go through some of the problems here. But the trial failures are what has been already talked about, these heterogeneous depressed populations. And we know that inflammation only occurs in a subgroup of depressed patients, about 30% on a good day. But these studies have been conducted on heterogeneous depressed populations by and large. Why would you expect to see an effect? The other problem is that the primary outcome variables, and Martine was kind of alluding to this, are non-specific measures of total symptom severity from the Hamilton Depression Rating Scale, the MADRS, the Montgomery-Åsberg Depression Rating Scale. If you've looked at those scales anytime recently, they got a ton of symptoms that have nothing to do with depression, and they're certainly covering the full gamut. But we know that inflammation has very specific effects. So when we start to talk about mechanism, we're going to start talking about specific effects on the brain and behavior that are not reflected in the outcome variables that we're currently using. And finally, the drugs that have been used, limited potency, multiple off-target effects. And so we really have to demonstrate target engagement. We have to show if we have a mechanism, we should hit that mechanism. We should hit that target. We should show that inflammation goes down and that that is in the blood or in the brain, and that that is related to behavioral changes. Now, I think most of you know that one swallow does not a summer make. So I'm not just blabbing on about inflammation here. We're not alone. This has already happened to us in psychiatry. You remember those drugs, the drugs that they developed for HPA axis, the hypothalamic pituitary adrenal axis? Years and years of research, preclinical research, went into studies showing that stress-related behavioral alterations in laboratory animals were related to activation of the hypothalamic pituitary adrenal axis. And a key molecule in driving that pathology in the animal models was corticotropin-releasing factor, CRF. So when I was a younger person, CRF was king, and the drug companies were coming out in droves. Everybody was developing their pet CRF antagonist, and it was going to change the world. People were buying stocks in these companies, and the drugs failed. And this is a review paper that was written several years ago. Don't stress about CRF. And it's a review about what happened. What were the failures? And this just gives you a feel, the number of drugs that were developed, the number of disorders that were looked at, and you can see pretty much across the board lacked efficacy. So in the paper, they go on to say, well, why do you think this happened? How was it that all this data, is all this data wrong? What happened that this was lost in translation from the bench to the bedside? And so they reviewed and they made what they thought was going on. And they said the efficacy of these drugs may be circumscribed to particular patient subgroups, to particular symptoms, to particular circumstances where you actually have activation of the CRF axis or the HPA axis. It's like deja vu all over again. Nobody's going to look at a CRF antagonist. And in fact, we may have thrown the baby out with the bathwater. We all know that these disorders are very heterogeneous, and that the point has been made several times. And I think that's one of the issues, one of the major issues, that if we don't change and we don't embrace precision psychiatry, this is where we're going to end up. You can see the majority of patients, CRF is normal, but there is 30, 40% in this particular study by Charlie Nemiroff, published in Science, that 30, 40% that have increased CRF. We knew this with inflammation a long time ago. This is almost 20 years ago. Look at this paper published in the American Journal that we published. You can see the majority of patients don't have increased inflammation. Give them all an anti-inflammatory and expect to get an effect. What are we doing? A little more in these are depressed cancer patients. Maybe the percent is. So depression is clearly not an inflammatory disorder. We have heard that one size does not fit all, and we all know that these populations that we call depressed or schizophrenic or whatever really represent subgroups of individuals with particular pathophysiologic mechanisms that are driving their symptoms. And we know a lot, and again, this is from last meeting where people were presenting data on what we know about how inflammation affects the brain. We know a lot about this, and we're going to hear a little bit more tomorrow. We're going to hear about what we know about immunometabolism, both at a cellular level as well as a systemic level. Brenda Pennings is going to talk about that tomorrow. So we know about this. We know about the inflammatory mediators that are involved. And Carmine Pariante is going to talk a little bit about this whole story here. And we know these inflammatory mediators can get into the brain, affect various neurotransmitter systems, dopamine, glutamate in particular, and that these changes in neurotransmitter metabolism affect neurocircuits in the brain that are related to motivation and reward processing, psychomotor speed and cognitive functioning, as well as threat sensitivity. And these manifest as specific symptoms and symptom domains that we can measure at a behavioral level. So the question then became, as we were working our way through this in this meeting in Switzerland, the psychiatry think tank, Precision Psychiatry think tank, is where do we go from here? And I think Marianne nicely touched on some of the conclusions. We're in the process of putting in a paper on the results of the meeting. And the strategies that we came up with are strategies that shouldn't surprise you. You need to enrich your populations, enrich for biological, clinical, or neuroimaging biomarkers. We're going to hear a lot about different biomarkers, different strategies of how to begin to deep phenotype patients and identify the patient that might be the best patient in terms of, in this case, looking at inflammation and its effects on the brain. So these could be inflammatory biomarkers. We're going to hear from Elizabeth Binder about genomics of risk and response. These are things that are one of a host of ways to enrich populations. But as both Martine and Marianne pointed out, you got to get past regulatory. If you're going to start stratifying people and you're going to start using biomarkers to identify patients and develop drugs to treat a particular disorder, this must be established, at least in the United States, within a context of use. And then there is a very arduous process of demonstrating reliability and validity of your biomarker. The advantage is if you get the right patients and you have the right drug, you may be able to get away with these kind of non-specific outcome variables, which is a whole issue unto itself. The second strategy that we talked about was developing novel clinical outcome assessments. And this really goes to what Martine was talking about, using scales that capture, in this case, inflammation's effects on the behavior. So moving away from these diagnostic categories and moving more towards domains. We're going to hear from Wayne Drevitz about scales for anhedonia. They've got a drug that hits a kappa opioid receptor, and they want to get anhedonia. In inflammation, anhedonia is a big target. But the challenge here is that, right, there's no you know, you have to get regulatory approval for any scales you use that isn't the Montgomery-Åsberg Depression Scale or the Hamilton Depression Rating Scale. There may be one or two others, but there's no scale for anhedonia that's been approved. And the amount of time and energy, and hopefully Wayne will fill us in tomorrow, Wayne. Interpret the results. So I think that the time is ripe, that there are mechanisms. I know where there was a sort of we need to know the mechanisms. I just gave you a mechanism. I just showed you that we have data, strong data, reproducible data showing that the immune system is affecting multiple pathways. And we know the pathways, we know the targets. So with intelligent clinical trial design and enriching patient populations, getting the right outcome variables, I think that this, at least in the case of inflammation, that we can lead the charge in precision psychiatry. And I started off talking about the costs. What happens if we don't embrace precision psychiatry? And that's what is, I think, leads me to the fierce.
So congratulations for all three speakers for setting the stage. I have a question for Andy when you're ready. So I don't know, maybe he's in the online audience. I don't see him. When Marion Boyer gave the keynote lecture at the EPA Congress, this was beginning of April, also on the topic of precision psychiatry, we got a very interesting comment and warning from Professor David Nutt, who was in the audience and who had been involved in the HPA axis research, which you've mentioned, and came back from that extremely discouraged. And so his warning to us was to not make the same mistakes and take lessons from the past. And so we talked to him after the lecture, and he explained to us that in this context of the CRF antagonists, they did actually speak with regulators. And at that time, was a long time ago, of course, they would not allow the use of any type of stratification measure to be able to select the patients for the trials. They basically told them flat out, we will never approve this because, and I hope that this is not true, but this is what he said, we do not believe that psychiatrists are capable of taking such a measure in clinical practice. So I'm really looking forward to hearing from regulators today. And I think it's extremely, extremely important that we convince them that psychiatrists today are very much capable of using biological measures in their practice. So how should we go about to do that?
Yeah, I'm sorry for David and the CRF story. I don't know what went on. I certainly was not in the room for the regulatory agencies, but I would imagine they probably were okay with a biomarker, but they probably said, you've got to do a lot of work to demonstrate to us that a biomarker is viable. And there are not many. If you go, I've looked on the website for the FDA. I haven't looked for the EMA, and we have representatives, and hopefully we can talk more about that tomorrow. How many biomarkers are really out there that people use? And one thing you should be aware of, if you develop a biomarker and you stratify, let's say you stratify on inflammation, and you have a drug that's an anti-inflammatory. If you're stratifying on the basis of a biomarker, you have to show your drug not only works in the high inflammation group, if that's what you're targeting, but doesn't work in the low inflammation group, which costs twice as much, potentially, depending on how you design the studies. So things get very complicated. And what we're talking about is multimodal deep phenotyping. So one biomarker versus many biomarkers. And Tyrone Cannon he got a letter of intent past the FDA. And I was told by the FDA, personal communication, I'm sure they'll deny it, but they said, we don't like these complicated biomarkers. We want something simple. We don't want these, complex biomarkers that include a number of elements in them to identify your target population. So we need to be careful. We need to think ahead. And that's why I think it's so good to have regulatory here and say, well, what's your appetite for a biomarker that includes 5 elements? And well, you got to validate each element and how they hang together. I don't know what they'll say. Be interesting. But it's a great question. And yeah, that CRH is a tragedy. And then you look with autism and oxytocin and the study in New England Journal of Medicine, the most ridiculous study I've ever seen. That's a field killer. Whoops. Is it up there? I don't know. Where did it go?
Well, it was a question from our colleague Lindquist from Sweden, who was asking. In the microphone, maybe for the. Who was asking Andy, what would be your best biomarker for CRF antagonistic study?
Well, I mean, that is something that maybe at the time they might not have had. I would probably ask Elizabeth to answer that question. Certainly you're not going to do a DEX CRH test, because it's now been shown the DEX test is probably not measuring what we thought it was. But there are, I think, strategies that could be used probably through genetics. Don't you think you could identify potentially a treatment responsive population with a combination of a genetic risk coupled with maybe an exposure, a gene by environment interaction? I think we've learned a lot. I think you could bring it back potentially. Maybe Florian has some on the back shelf that he left there, and bring it back out once we get more on that. But Elizabeth is going to talk more about that tomorrow, and that's what I would be thinking, Carmine.
I want to ask you a question, just bringing back to, if you like, combining future and past psychiatry. And I wonder if you think there's any space for research on predictors of treatment that are already available. So in other words, we still don't know why some people respond to SSRIs or tricyclics, or I guess with the newer, ketamine or combination with that typical. And I mean, obviously, like Andy, I'm passionate about the immune system, and I believe that, immunopsychiatry is definitely a frontier for the future treatment for that particular subgroup. But what about predicting response to the drug that we already have? Lithium versus tricyclic or SSRIs, and yeah. So of course, I'm aware of there has been a lot of literature, but is there a recurring theme? Is there something that hasn't been done that could be done within that space?
Well, there was a review in World Psychiatry where they went through a series of biomarkers that are out there, and most of the biomarkers that are out there right now are used to go for a diagnosis, a prognosis, or a treatment response to a traditional agent. And the NIH in the United States is funding studies to predict response to traditional, conventional pharmacologic interventions, or even behavioral interventions. It doesn't matter. So there is interest in it. Again, my interest is more the targeted treatments, and so that you're just predicting response to the standard out there. It's just not my thing. It's not making a judgment at all. It's just not. We're trying to develop novel drugs.
So it may be an interesting way to maybe find biologically homogeneous clusters to understand why that group is responding or not. That could be one way of using it, but there may be better ways of actually coming to these more homogeneous clusters by using truly quantitative biological assessments. But clearly, some people are responding, others are not. So that is telling us something about the biological variation in those groups.
Do you think there's a kind of overlap between some of the older concepts and the new concepts? So, for example, is it possible that inflammation could predict response to a particular type of tricyclic or a particular type of SSRIs? I mean, there's some data that venlafaxine has a stronger anti-inflammatory action, at least in vitro in animals. You know, so is there something that we could almost kind of overlap, or we, you know, the future version and the past version, so that we can use some of the biomarker analysis not only for obviously target engagement of a proper anti-inflammatory medication, but also for medication that were not developed as anti-inflammatory, but maybe they do work as anti-inflammatory as well.
I think Brenda wants to respond.
Yeah, I think one problem with addressing that question is the data. Do we have the data to do this? And I think if you just look at how many trials of antidepressants do we have, there are lots of antidepressant trials. How many of the, and some of these are really historical trials. How many of these trials have collected blood? Then you're reduced to probably 10 studies. And I think there are a few indications in the literature out there. And we analyzed actually 4 larger, I think I have even a slide on it tomorrow. We analyzed some of the larger trials testing just the simple SSRIs, looking at whether CRP, for instance, in combination maybe even with BMI or with some other indicators, are predicting treatment response. It's only really limited. It is predictive, but really to a limited extent. But it's amazing how little data we have to address these kind of questions, because lots of trials till, you know, a few years ago didn't really collect systematic blood samples.
Hi, Anneli from Olink Proteomics. I think this is a very interesting discussion. I just wanted to comment from our perspective. We're seeing an increased appetite from pharma companies to actually go back and look at failed clinical trials, to look deeper, for example, with a proteomic method, but also with other methods to better understand responders and non-responders, why it failed overall. So that's what we're seeing.
Brent, sorry.
I mean, just one comment. I mean, in the genetics field, there's a big controversy about also genetic risk factors. Is it basically everybody has their own personal genetic risk, or is there convergence on certain pathways? And there are strong arguments against convergence on certain pathways. And the question is, how likely will we actually find these subgroups that we can target? And maybe also a question to like immunopsychiatry, like I understand that we have a third who have elevated immune markers, but is that causally related to the symptoms, or is that a correlation, and would actually changing that in all of the patients who have higher inflammation lead to that? So I think it's very exciting to think about that, but I also feel that we have a big gap in understanding mechanisms in psychiatry so far.
I would make a comment about that, but I won't. But I know I can't resist. So there's, I think, very strong data of cause and effect relationship between inflammation and depression. Data from our lab with infliximab. We're just about to publish another paper on infliximab that really drills down with what exact brain regions. It all fits with the anhedonia story and whatnot. And it's very clear that if you give people interferon or an inflammatory cytokine, they get depressed. So you can go either way. Neil Harrison did a nice study with interferon and infliximab as well. So there's good data out there, I think, to say that there's a cause and effect relationship. The poor performance of the clinical trials, you know, it may be that they're, you know, we're just not using the right drugs or whatever it is. But, you know, I do think there's enough data out there to suggest that this is this is the real deal. So I couldn't resist. I'm sorry.
If I may comment to the question, I'm agreeing with Andy on the causality, and also I'm hearing over and over again, we don't understand the mechanisms enough. We need to do more research into the mechanism, but we cannot ever understand the mechanisms if the diagnostic categories are incorrect. So it's a chicken and the egg question. If we do not move away in our biological studies from using DSM-specific categories, we're not going to find the mechanisms, and then we're not going to find better treatments. So it's, yeah. How do we get out of this situation?
Thank you, Livia. So maybe before we end this introduction session, we have a few questions from the online colleagues. And one is a general question that needs to be addressed, I think. The question is, what will be your advice for those who are living in Africa, who are far behind even in the area of few validated diagnostic tools? Investigations are still basic. So we very often hear this type of criticism that is going to be costly and difficult to be, enrichment is going to be costly. So you want to answer, Martin?
I think it's a fair point, right? I mean, initially we probably need expensive technology to figure it all out, but at some point we will need proxies that are scalable and allow us to also have other countries that have maybe less money to do so to follow the same path. But that's a long way forward. I think we first need to know, probably with more expensive technology, the biology, and need more cheaper, scalable proxies to address that. I think that will probably be the path.
If I just, I mean, I totally agree. And if I just can add to that, we had in the last few months discussion with the Wellcome Trust. We finally got awarded a big grant, some of the PIs are in these rooms. And obviously the Wellcome Trust has always been very interested in translating the research findings across the whole world. And immunopsychiatry is a particular area which could be developed within kind of different settings, because of course immune laboratories are available everywhere in less developed country because of the importance of infectious disease in the context of their local health. So there maybe there's an opportunity with at least with immunopsychiatry and some of these biomarkers, not the more mechanistically sophisticated, but some of the one that can be assessed at the hospital level, really to be implemented quite rapidly in some of the countries where, as the colleagues said, in Africa or similar. So, it's not all, it doesn't always have to be really costly, and mechanistically could already be clinically relevant, maybe translatable from other aspects of physical health. Yeah.
Yes, good point. For certain fields, we really have markers which are in the public domain already. It could be used once it's validated by clinical research, which are expensive, but not the clinical applications. At least we can hope. So if you agree with this positive answer and hope, we can stop here for this evening, and thank you very much for your attention and your contribution, and thank you very much to our online colleagues who've stayed a long time despite difficult, I think, connections. Thank you. I hope we're going to have a nice dinner now, but they're not going to have it. Yeah, absolutely. Thank you very much.